Okay, so for this video, we are going to start with antibiotic resistant mechanisms. We are doing basic bacteriology in microbiology module. This is a bacterial cell. It has ribosomal RNA and then messenger RNA that form the ribosome, and this is producing an enzyme from the DNA. And these are the proteins these are the gated channels or non-gated channels in the cell membrane wall, and these are also some proteins that are present. So the first mechanism of antibiotic resistance is enzymatic inactivation. Um, there is drug cleavage by this protein, cleavage of specifically penicillin G, penicillinase sensitive penicillins, cephalosporins, and carbapenems. The drug modifications are done for aminoglycosides and chloramphenicol inside the cell. So when you give these drugs, they are basically cleaved, these penicillins and cephalosporins. And when you give aminoglycosides and chloramphenicol to these resistant organisms, they are just modified. The second mechanism is increased efflux. This drug is rapidly moved out and decreased uptake. It is for tetracycline, sulfonamides, and fluoroquinolones. Then we have drug target modification. For example, if the drugs target ribosomal RNA as linezolid and macrolides, it is inhibited there. If it targets specific enzyme, for example, penicillinase resistant penicillins, okay, so we have discussed penicillinase sensitive penicillins that work basically by enzymatic inactivation, but penicillinase resistant penicillins, they are modified by the inhibition of the enzyme they act on. Then we have cephalosporins again, and sulfonamides and fluoroquinolones again. Um, they they are provided resistance by drug target modification. Sulfonamides, fluoroquinones are also by increased efflux and decreased uptake. And then we have cell wall on which vancomycin acts, and the cell wall will inhibit the action of vancomycin to provide antibiotic resistant mechanisms. Then we have bacterial genetics. It is the horizontal gene transfer, which is the main mechanism for transfer of antibiotic resistance among bacteria. The first is transformation. Okay, there is naked DNA, and this is a recipient cell. It has taken up this naked DNA and degraded the uncombined DNA and taken up some of the naked DNA and added it to um, the donor DNA, and now the cell is transformed. And there's also some of the degraded uncombined DNA. Competent bacteria can bind and import short pieces of environmental naked bacterial chromosomal DNA from bacterial cell lysis. The transfer and expression of newly transferred gene is called transformation. Transform cells. Now I'm transformed. A feature of many bacteria, especially S. pneumonia, H. influenza, and Neisseria again. And these were the encapsulated bacteria, if I remember well, and they were also present right here having IgA proteases. So these three bacteria had this thing in common that they are bacteria that undergo transformation. They are bacteria that are encapsulated, and they are bacteria that have IgA protease. Adding deoxyribonuclease degrades naked DNA, preventing transformation. So if you add a ribonuclease, deoxyribonuclease, it degrades the naked DNA and prevents this transformation, which is important clinically. Then we have conjugation, um, an F positive and F negative conjugation. An F positive is a fertility factor plasmid that contains genes required for sex pilus and conjugation. Okay, let's look at the diagram first. This is an F positive cell. It has F positive plasmid that contains genes for sex pilus and conjugation. Then we have an F negative cell, and the sex pilus forming conjugal bridge or mating bridge. This sex pilus forms a conjugal bridge right here. And then this F positive cell transfers its F positive plasmid, single strand of the plasmid DNA transfer to the F negative cell. There's no transfer of chromosomal DNA, it remains separate, and both the cells are now F positive. Bacteria without the plasmid are termed F negative. So, what determines the fertility factor is the plasmid. If there is a plasmid, we're going to call it F positive, and if there's no plasmid, we're going to call it F negative. Sex pilus and F positive bacterium contains F negative bacterium, contacts F negative bacterium. A single strand of plasmid DNA is transferred across the conjugal bridge or the mating bridge, and there is no transfer of chromosomal DNA. We then have HFR across F negative. So again, there's an F positive cell and an F negative cell, And then we have a high frequency recombination cell that contains F positive plasmid incorporated into bacterial DNA. So this was a separate plasmid and separate bacterial DNA, but in an HFR cell or a high frequency recombination, the plasmid recombines with the DNA and there is an F negative cell. Now, sex pilus forms and the leading portion of plasmid transfers along with flanking bacterial chromosome. There is a transfer of leading portion of plasmid as well as bacterial, a part of bacterial chromosome. And then there's an HFR cell and recombinant F negative cell because there's going to be no plasmid additionally, it is just going to add into the DNA. So F negative cell bacterial DNA plus F positive cell bacterial DNA plus plasmid copy is present in here. And there's no plasmid, so it remains F negative and does not become positive. F positive plasmid can become incorporated into bacterial chromosomal DNA, termed high frequency recombination cell. Transfer of leading part of the plasmid and a few flanking chromosomal genes. High frequency recombination may integrate some of those bacterial genes. Recipient cell remains F negative, but now may have new bacterial genes. So both of these are part of conjugation because there is sex pair is forming and this was transformation. Now we have transduction. Here we have a bacteria that has a lytic phage or a bacteriophage that causes its DNA to enter, and then there's the cleavage of the bacterial DNA. This phage causes cleavage. Bacterial DNA packaged in phage capsids. So the phage releases capsids, and the bacterial DNA that was lysed is packaged here, and release of new phage from the lysed cells. So new phage gets released and infects other bacteria, and genes transferred to new bacteria from this phage. Generalized is a packaging error. Lytic phage infects bacterium, leading to cleavage of bacterial DNA. Parts of bacterial chromosomal DNA may become packaged in phage capsid. Phage infects another bacterium, transferring these genes. Specialized would be a lysogenic bacteria right here. Okay, so generalized transduction is caused by lytic and specialized is caused by lysogenic. The viral DNA enters and incorporates in bacterial DNA. Phage particles carry bacterial DNA and release of new phage particles form lyse cells and infect other bacteria and genes different from donor and recipient. So it is completely new organism that is being formed here. It is an excision event. Lysogenic phage infects bacterium, viral DNA incorporates into bacterial chromosome. Okay, there was no, in the lytic phage, there was no incorporation of the viral DNA in the bacterial chromosome. It was just cleavage of the bacterial DNA and packing into the phage capsid. This one actually causes the incorporation of viral DNA into bacterial chromosome. When phage DNA is excised, flanking bacterial genes may be excised with it. DNA is packaged into phage capsid and can infect other bacteria. Genes for the following 5 bacterial toxins are encoded in a lysogenic phage. Group A strep erythrogenic toxin, botulinum toxin, cholera toxin, diphtheria toxin, and Shiga toxin. All of these are encoded in a lysogenic phage, these ABCDs. A strep erythrogenic toxin, B botulinum toxin, cholera toxin, diphtheria, and then again Shiga toxin. Then we have transposition. This is a plasmid that has a portion which is known as the transposon. And the transposon in the bacterial DNA has a target site. And at this target site, this transposon comes, this part, and attaches and causes integration of genes. So transposition is a jumping process involving a transposon, which is a specialized segment of DNA which can copy and excise itself and then insert into the same DNA molecule or an unrelated DNA, example, plasmid or chromosome, critical in creating plasmids with multiple drug resistance and transfer across species line. Example, TM1546 with vancomycin A from Enterococcus to S. aureus. Okay, so Enterococcus has vancomycin A resistance and it transfers that to Staph aureus with the help of transposition, and this causes multiple drug resistance and transfer across species line. Then we have main features of exotoxins and endotoxins. Okay, so this is an exotoxin. It is being released and has a downstream cellular action in the host cell. This is an endotoxin that is basically present in the cell wall, as we discussed for LPS, and then it goes and binds to a receptor, and then it causes TNF, IL-1 or IL-6 expression in there. So, exotoxins, the source is certain species of gram-positive and gram-negative bacteria. Endotoxins are mostly from the outer cell membrane of gram-negative bacteria because that contains LPS. There are exotoxins secreted from cells and endotoxins are not. Exotoxins are composed of polypeptides and endotoxins are comprised of lipid A component of LPS, and it is released when lysed because it is structural part of bacteria. The location of genes for exotoxins is in the plasmid or bacteriophage, and endotoxins location of genes is in the bacterial chromosome because an endotoxin is basically a part of the bacteria. Toxicity is high, fatal dose on the order of 1 microgram. Exotoxins are highly fatal, endotoxins are low, fatal dose on order of hundreds of micrograms. Clinical effects are various effects, and there are various modes of action of exotoxins. Endotoxins cause fever, shock, and DIC, and their mode of action is inducing TNF, IL-1, and IL-6. Antigenicity for exotoxins, they induce high titer antibodies called antitoxins, and endotoxins are poorly antigenic. Vaccines, toxoids are used for vaccines for exotoxins, and no toxoid form and no vaccine available for endotoxins. Heat stability, they are destroyed rapidly at 60 degrees Celsius except staph enterotoxin, E. coli heat stable toxin, and BCA schematic toxin. And endotoxins are more stable, and they're stable at 100 degrees Celsius per hour. Except staph enterotoxin, E. coli stable toxin, and B. serious emetic toxin. Heat stability is poor for exotoxins. Then we have typical diseases associated with exotoxins. These are tetanus, botulism, diphtheria, and cholera. And with endotoxins, we have meningococcemia, and there's sepsis by gram-negative rods caused by these endotoxins. Okay, moving on. We have bacteria with exotoxins. The first bacteria are those that inhibit protein synthesis. First one is Corynebacterium diphtheria. And the second one is Pseudomonas aeruginosa. Their toxin is called exotoxin. They secrete diphtheria toxin, and Pseudomonas secretes exotoxin A. They inactivate the elongation factor through ADP ribosylation. So the elongation factor is a factor in the bacterial transcription or DNA. I think it was a factor in DNA translation, and its elongation factor causes elongation of the um, um, elongation of the. Let's just search it up. Elongation factor. EF2. Involved in translational elongation process. It facilitates the translocation of ribosome during translation, moving the peptide to transfer RNA from A site to P site of the ribosome. So both Corynebacterium diphtheria and exotoxin A, they inactivate this elongation. And pharyngitis with pseudomembranes in throat and severe lymphadenopathy and myocarditis. So when they inhibit this elongation, they can the Corynebacterium diphtheria can cause pharyngitis with pseudomembranes in throat and severe lymphadenopathy called a bullneck. I want to see a bullneck. Images. So there is inflammatory exudate or pseudomembrane appearance in the pharynx, and there is a bullneck right here because of cervical lymphadenopathy caused by this diphtheria toxin. And we have exotoxin A that causes host cell death. And Shigella and enterohemorrhagic E. coli have Shiga toxin. Enterohemorrhagic. They inactivate the 60S ribosome by removing adenine from the ribosomal RNA. So the 60S ribosome is the component of the greater component of the ribosome, and these sugar toxin inactivates this ribosome and removes adenine from the ribosomal RNA. It damages GI mucosa and causes dysentery and toxin-mediated injury and cytokine release in the hemolytic uremic syndrome, typically in EHEC serotype 015787. Unlike Shigella, EHEC does not invade host cells. So it is going to cause dysentery in the GI mucosa and hemolytic uremic syndrome in the body. And if this HUS is typically caused by EHEC serotype 015787, that does not invade the host cell unlike Shigella. These were the bacteria having exotoxins that inhibited protein synthesis. We are now going to do bacteria that increase fluid secretion. We have enterotoxigenic E. coli um, that have a heat labile toxin and a heat stable toxin. So the heat labile toxin overactivates adenylate cyclase, a cyclic AMP, and that increases the chloride secretion in gut and water efflux, and watery diarrhea, labile in the air, stable on the ground, guanylate cyclase. Bacteria that increase CAMP include Vibrio cholera, B. anthracis, B. pertussis, and E. coli. So all of these increase the CAMP, and this they cause increased fluid secretion and their heat labile toxins. Then it also has a heat stable toxin, this E. coli, enterotoxigenic E. coli. It overactivates CGMP and decreases the resorption of sodium chloride and water in gut, which causes increased fluid secretion again. So stable on the ground, guanylate cyclase, labile in the air, adenylate cyclase. And again, the bacteria that increase CMP include Vibrio, botulinum, pertussis and anthracis, and E. coli. Then we have, Bacillus, not botulinum, Bacillus. Then we have Bacillus anthracis that obviously has anthrax toxin. It mimics adenylate cyclase. It is likely responsible for characteristic edematous borders of black esper in cutaneous anthrax. Okay, so anthrax is basically responsible for anthrax and it increases the fluid secretion. So it is responsible for edematous borders of the black escarine cutaneous anthrax. Then we have Vibrio cholera that has a cholera toxin that overactivates adenylate cyclase by permanently activating the stimulatory protein. And there's voluminous rice water diarrhea with Vibrio cholera, obviously. Again, we had those that inhibit protein synthesis, Corynebacterium, Pseudomonas, Shiga toxin, and we have increased fluid secretion because of a heat stable, heat labile, anthrax, and cholera toxins. Now we have to inhibit the phagocytic ability with Bordetella pertussis that secretes the pertussis toxin. It activates adenylate cyclase by inactivating inhibitory subunit. So it acts kind of similar to Vibrio cholera, but it inactivates the inhibitory subunit, and Vibrio was permanently activating the stimulatory subunit. Whooping cough is because of Bordetella pertussis, child coughs on expiration, and whoops on inspiration. It can cause 100 days cough in adults associated with post-tussive emesis. Okay, whooping cough. Important clinical point, by the way. Then we have the bacteria that inhibit the release of neurotransmitter. Clostridium tetani causes the secretion of tetanospasmin, and Clostridium botulinum causes the secretion of botulinum toxin, obviously. Both are proteases that cleave snare soluble NSF attachment protein receptor, a set of proteins required for neurotransmitter release via vesicular fusion. So yes, snare protein is an important protein that is required for neurotransmitter release, and tetanospasmin and botulinum toxin are both going to cleave the snare. Tetanospasmin prevents the release of inhibitory GABA and glycine neurotransmitter from ventral cells in the spinal cord. Okay, and this can cause spastic paralysis, rhizus, sardonicus, trismus, and opus thotonos. So there's a lot of terminology to be googled here. Rhizus sardonicus. Okay, basically spasm of facial muscles is rhizus sardonicus. Then we had trismus or lockjaw. Oh, a lockjaw. Okay, so the jaw presents like that. And then we have opus thotonos. Opus thotonos. Opus thotonos. Uh, Abnormal posturing caused by, okay, so the person presents like this, all because of Clostridium tetani and the tetrospasmin that causes spasm and by inhibition of GABA cells in the inhibition of GABA neurotransmitter and glycine neurotransmitter from ventral cells. GABA and glycine are, I think, inhibitory neurotransmitter. I mean, there's no inhibition for the ventral cells in spinal cord. There's going to be spastic paralysis, rhizus sardonicus, which is again the facial muscle paralysis, trismus, which is lockjaw, and opisthotonos, which is an abnormal posture of the body because of increased spasm. Then we have Clostridium botulinum. It causes infant botulism, which is caused by ingestion of spores, example from soil, raw honey, and toxin produced in vivo. And then we have foodborne botulism, that caused by ingestion of preformed toxins from canned food. Okay, so it is causing botulism either way, infant botulism and foodborne botulism. An AB toxin, also called two-component toxin, or 3 for anthrax, with B enabling binding and triggering uptake of the active A component. The A components are usually ADP ribosyl transferases. Others have enzymatic activities as listed in chart. So all these in diphtheria toxin, enterotoxin, Shiga toxin, sorry, exotoxin A, Shiga toxin, heat labile toxin, not heat stable, anthrax toxin, cholera toxin, pertussis toxin, hypnospasmin, and botulism toxin are AB toxins, and they have two components. Only 3 for anthrax. The B enables binding and triggering uptake of the active component. Uh, the A components are usually ADP ribosyl transferases, and there's have enzymatic components is listed in the chart. The only others that are left were heat stable toxins, and all of the rest had an A component and B component, with the A component usually being ADP ribosyl transferase. Now we have bacteria with exotoxins continued, and these bacteria now lyse the cell membrane. We have discussed bacteria that inhibit protein synthesis, that increase fluid secretion, that inhibit phagocytic ability. And then we have inhibition of release of neurotransmitters, and now we have lysis of cell membranes. Clostridium perfringens and Streptococcus pyogenes. So Clostridium has alpha toxin, and Streptococcus has streptolysine O. Alpha toxin causes phospholipase or lecithinase that degrades tissue and cell membranes. There's degradation of phospholipids that causes myonecrosis or gas gangrene and hemolysis, double zone of hemolysis on blood agar. Okay, so this is very important and very interesting that Clostridium perfringens has an alpha toxin, and this alpha toxin is basically an enzyme kind of. It has an enzymatic phospholipase kind of action that degrades the tissues and cell membranes. And this degradation of phospholipids causes myonecrosis, which is known as gas gangrene, and hemolysis, which would be shown by double zone of hemolysis in blood agar. Then we have Streptococcus pyogenes that has streptolysin O as an exotoxin. It is a protein that degrades cell membrane, streptolysin O, and it lyses RBCs, contributes to beta hemolysis, host antibodies against toxins, used to diagnose rheumatic fever, ASO. Do not confuse with immune complexes of PSGN. Okay. So, ceftriaxone O is contributing to beta hemolysis, and the host antibodies against the toxin ASO are used to diagnose the rheumatic fever. This is particularly clinically important that these host antibodies against toxin ASO are used to diagnose rheumatic fever because um, it would be an antigen against ceftriaxone O, which would mean that there is ceftriaxone in the body, and you would detect that there is rheumatic fever, which is caused by step pyogenes. Then they have super antigens causing shock. These have Staph aureus and Streptococcus pyogenes. Staph aureus has toxic shock syndrome toxin, TSST1, that cross-links and Streptococcus pyogenes has erythogenic exotoxin A. Not only did it have streptolysin O, it also has erythogenic exotoxin A, and both of these cross-link the beta region of um, T cell receptor to MHC class 2 on APCs outside of the antigen binding site. This causes overwhelming release of IL-1, IL-2, IFN gamma, and TNF alpha, and that causes shock. So they cross-link the beta region of the receptor complex to MHC on APCs outside of the antigen binding site. And this increased cross-linking of the TCR to the MHC causes obviously increased inflammatory response and increased interleukins 1, 2, IFN gamma, and TNF alpha. Toxic shock syndrome, fever, it has fever, rash, shock. Other toxins cause scarlet skin syndrome, exfoliative toxin, and food poisoning, which is heat stable enterotoxin. So Staph aureus has toxic shock syndrome toxin as an enterotoxin. It also has an exfoliative toxin that causes scarlet skin syndrome, and a heat stable enterotoxin that causes food poisoning. And toxic shock like syndrome is caused by Streptococcus pyogenes. It has fever, rash, shock, and scarlet fever. And scarlet fever is a fever that has a strawberry shaped tongue presentation, pharyngitis, and impetigo, I think. Then we have endotoxins. Endotoxins are, as we have discussed in the previous part, LPS found in outer membrane of gram-negative bacteria, both cocci and rods. They are composed of O antigens plus core polysaccharide plus lipid A, the toxin component. Neisseria have lipo-oligosaccharide. So Neisseria are such fussy organisms. They are gram-negative, but they have lipo-oligosaccharides, and I think they also required the um, thiamine or Theramere, whatever it was, for their mm, yes, Thermartin. This area required Thermartin because it's liquid flavors this area. And here it has an exception that it has lipooligosaccharide instead of just lipopolysaccharide. Um, Releases the endotoxins are released upon cell lysis or by living cells by blebs detaching from the outer cell surface membrane versus exotoxins which are actively secreted. There are three main effects: macrophage activation of the toll-like receptor 4 or CD14, complement activation, and tissue factor activation. Endotoxins would cause E for edema, N for nitric oxide, D for DIC or death, O for outer membrane activation, T for TNF alpha, O for O antigen plus core polysaccharide plus lipid A, which are extremely stable, I for IL-1 and 6, N for neutrophilic chemotaxis, and S for shock. So, this is a very pretty chart that explains that lipid A component of endotoxin causes macrophage activation by binding to TLR4 and CD14 that causes release of IL-1 and 6, the feverish interleukins, TNF alpha that causes fever and hypotension, and nitric oxide that causes hypotension. Then it causes complement activation of C3A and C5A, and C5A also activates C3A, and this causes histamine release. And the histamine release is accompanied by hypotension and edema. And C5A also causes neutrophil chemotaxis. Then it causes tissue factor activation that causes the coagulation cascade and DIC again. So there's going to be fever, lots of hypotension, neutrophil chemotaxis, and DIC. Okay, and we will touch some part of this clinical bacteriology before we begin with bacteria in our next video, and this would be this page. Gram-positive lab algorithm would be a gram-positive bacteria is purple or blue, and they are classified into bacilli, which are rod-shaped, cocci, which are these circular bacteria, and branching filaments, which are basically like the filaments or like branching branches of a tree. The bacilli, the rod shape, are classified into aerobic and anaerobic. The aerobic is Listeria bacillus and Corynebacterium. And the anaerobic is the Clostridium, Cutibacterium, formerly known as Propionine bacterium. This is such a nice name. I've just discovered this one. Anaerobic bacteria is called Cutibacterium, and it was formerly called Propionine bacterium and aerobic bacilli, by the way. Then we have cocci, which are catalase positive or catalase negative. The catalase positive is Staph, which is present in clusters, and these Staph can be coagulase positive, this is Staph aureus, or coagulase negative, which have novobiasine sensitivity, and these no. So the coagulase negative could either have novobiasine sensitivity, Staph saprophyticus, or um, it could either not have it, as in case of saprophyticus, and it could have it as in case of epidermidis. Then we have the catalase negative organisms, which are the streptococcus. Pair of chains are not clusters. They are classified on the basis of hemolysis. Alpha hemolysis is partial hemolysis. It is green, and it is optochin sensitivity and bile solubility. Uh, Optochin sensitivity. I have no idea what that is. Optochin sensitivity. Lab tests used to presumptively identify strep pneumonia by observing its susceptibility to ethyl hydroxyprene hydrochloride. Okay. So it is basically a molecule used to uh, separate S pneumonia, which is encapsulated from viridans and S mutans or S mutis. Then we have beta, which have besitracin sensitivity and PYR status. And on the basis of this besitracin sensitivity, if it is negative, it is going to be group B. And if it is positive, it is going to be group A. Then we have gamma hemolysis, that is no hemolysis and it grows in bile. It grows in 6.5% NaCl and PYR status. If it grows in 6.5% NaCl and it is a gamma hemolysing Streptococcus, it is going to be Enterococcus. And if it does not grow in 6.5% NaCl, but it is a gamma hemolysis strep, then it is going to be non-Enterococcus S. Galoleticus. Galoleticus. Yes, that's what it is. Then we have branching filaments, which are very cute. They have aerobic and anaerobic. Aerobic are going to be called nocardia, which are weakly acid fast, and anaerobic are going to be called actinomyces, which are not acid fast. So this was it for this part. We will continue in the next part. See you then. Bye.